areas of specific specialization is microbiology, and marine biotechnology, and aquaculture. I don't know what aquaculture is, but it will help them. Okay, let me introduce her career briefly. So she graduated the University of Vigo in Spain, 1986. And then she entered PhD course in the University of Santiago de Compostela. And then she received PhD in 1994 in the Spain University. And in 1994, she visited Japan for 2 months in NIDB, that is the National Institute for Basic Biology. The laboratory headed by Professor Murata, and she stayed there for 2 months. And then she became postdoc in the University of Santiago de Compostela. And in 1996, she came to Japan again as EU, European Union, postdoctoral fellow, and she stayed in Japan for 2 months as a postdoc. That is 1996 and 1997, and then she came back to Spain, and she got assistant professor position in 1998 in University of Colonia. And in 2000, she became assistant professor in the University of Santiago de Compostela. And in 2004, she became associate professor in the same university. So 18 years back, so Anna-san and I were working in the same laboratory. And 15 and 60 years ago, she was working in the same laboratory as a postdoc. At the time, the laboratory was called Laboratory of Stress Response of Plant, but it's a long name, so we call it simply stress now. And that laboratory is to understand the stress response of plant, but people are very much stressed, and only the people who who are resistant to stress could survive, and she is one of them, and I would say she's quite stress resistant lady. Very not Okay, so we have been friends more than 16 years. So this time, we invited her as a lecturer for this high-set program. So today, she has the first lecture, and Friday morning, she's going to have a lecture for first grade of undergraduate student. And in the afternoon, she has the third lecture for scientific presentation for second and third grade of the undergraduate student. So it's the time to finish the introduction. Okay, Anna-san?
So, good afternoon, everybody. First of all, I would like to thank the professors in charge of the HiSEP program for inviting me, and especially my good friend Nishiyama-san. For many years, that was the one in charge of bringing me back to Japan after so many years. I'm going to talk to you this afternoon about bacterial communication, and more specifically how we can take profit of bacterial communication. But before talking about bacteria, I would like to introduce a little bit myself beyond the scientific profile that Nishiyama today has done. So first of all, I would like to explain where I come from. I come from Spain, and the city is called Santiago de Compostela. When I say I come from Santiago de Compostela, most people said, oh, that's in Chile? Santiago de Chile? I said, no, that's in Spain. This is the original Santiago. All the other Santiagos in South America are named after this Santiago. That was a very important middle-aged city in Spain. It's located, as you see, on the north of the country, very close to Portugal, and is facing the Atlantic Ocean. That means that we have a lot of rain. All the rain that comes to Europe from the Atlantic Ocean comes through Galicia, through my region. So the landscape is very different from the normal Spanish landscape. And many other things are different in Galicia. So first of all, it's green. In Spain, they call us the land of 1,000 rivers. So because it's very, very rainy. We don't dance flamenco. Our folk music is related to Irish music, so we play bagpipes. And we have a very, very nice coast that is very similar to the Norwegian coast. And that makes us the second world producer of aquaculture mollusks, mainly mussels. So you can see here, these are the facilities where we cultivate the mussels. So after China, we are the first world producers for mussels and other seafood. Santiago de Compostela is a very famous city, especially in Europe, because it's a holy city for Catholics. We have a very ancient cathedral that was built in the 11th century. The reason why it's holy for the Catholics is because it's a tomb of St. Jacques, which is, there are three holy cities for Catholics, which are Rome, Jerusalem, and Santiago. And Santiago is holy because it's the only person that is known where the tomb is that met Jesus Christ. That's the reason. So during the Middle Ages, if people could not go to Jerusalem, they came to Santiago, walking from all Europe to Santiago de Compostela, and they still do nowadays. So every year we get something like 300,000 pilgrims that come walking from all over Europe to Santiago. It's a very touristic place. And the second most important thing is the university. So it's the second oldest university in Spain. It's a public university. It was founded in the 15th century. We have 2 campuses and 23 faculties with more than 40 degree courses and around 2,000 professors for 27,000 students. So it's a quite large university. It's a 5th university in Spain. Regarding research in our university, we are especially strong in life sciences. So recently we have been awarded, what you see here is Campus Vida, means campus life. We are among the 10 top universities in Spain regarding research in life sciences. So my PhD was, I did my PhD in that university, and I work in a very different thing from what I'm going to talk today. I work on microalgae biotechnology. The purpose was to study the physiology of microalgae in very dense cultures that are producing this kind of machines that are called for bioreactors. At that time, the purpose was to produce fine chemicals like astaxanthin. This is astaxanthin from Matococcus, or microalgae to feed animals, marine animals, and most importantly, to produce long-chain polysaccharide areas. Because the marine eukaryotic microalgae are the only, almost the only microorganisms that produce these very unique substances. So that was the reason why, after my postdoc, I joined the stress lab in NIBB. So I was extremely lucky that I was invited. It was very, very hard, but I think that it was probably a unique opportunity to meet excellent researchers, very well known and renowned all over the world. So you have here Nata Sensei, and Toby G, and Papa Giorgio, many important things were there at that time. it was a great opportunity. So, I'm going to talk today about a completely different thing, and you may be thinking, what's the link between microalgae and quorum sensitive bacteria or communication bacteria? Well, no link. When I finished my postdoc in Japan, I took another postdoc in Italy, and finally I got a position in Spain, but at that time there was no money for microalgae, so I had to think of something new. That was a pity, because only 3 or 4 years later, we attended to this in Europe and all over the world. So microalgae are again at the top for getting money due to the biodiesel high. And many researchers and companies get interested in the production, the use of 4 bioreactors for the production of biodiesel. But I'm almost out of the field. We still have in our laboratory a group of people assisting 4 bioreactor operation in some companies and research groups, but that's not anymore my main subject. So if I have to blame somebody of quitting the algae field and entering the bacterial communication field, it's this lady. So in 2002, I met this, attended a conference and watched her presenting the result about multilingual bacteria, and she changed my life. She's probably the best scientific communicator I've ever seen, so I very much advise you to watch her talking in this in TED website, and she is really fantastic. I really advise you to see her. So Bonnie Butler was working on bacterial communication at that time, and when she finished her lecture, I said, I want to work on that. I was absolutely fascinated. It took me some time to get there, but I did it. And before listening to Bonnie Butler, my idea of bacteria is probably the idea of most researchers have, a very boring thing. And this is François Jacob. She's a Nobel Prize. In 1973, he said this about bacteria. Very boring thing. They can adapt a little bit to the environment, try to survive, and that's all. They can do much more. But even at that time, 1973, we already know that bacteria had developed very refined ways to communicate. And they were not boring at all. And that was discovered in these unique marine bacteria. These are bacteria from the genus Vibrio, and they produce light. So many marine bacteria, marine biotechnologists and molecular biologists got interested in these bacteria at the end of the 70s and at the end of the 60s and 70s. And what they discovered is that these bacteria do not produce light continuously. So if you have a culture of these bacteria, this is what you see. This is growth. Growth is continuous, logarithmic. But light is not produced at the beginning. When the density is very low, almost no light is produced. And then all of a sudden, the bacteria agree to produce light in a synchronous way, all at the same time. And that was very weird thing, very strange. How do they know when they have to start producing the light? So they made that experiment at that time, that was late 60s or very early 70s, and they realized that there was a signal that is being released to the culture media, and when a threshold concentration of the signal was achieved, then all bacteria knew that they had to start making light together. That was very unique, because at that time, most scientists thought that only eukaryotic cells could communicate, have hormones to communicate, and differentiate. For bacteria, nobody thought it was possible. They said, no, it's not possible. This is very unique for these bacteria. A few years later, they discovered the molecule, the signal that these bacteria were producing. This is an acetylmosylate lactone. It has a lactone ring that comes from the some metabolism, some molecule, and a fatty acid tail. And the community, most of all the scientific community knew this result, but because at that time a lot of people was very interested in luciferase, but they didn't pay much attention. They said, okay, this is very unique to this bacteria because of they are very specialized. Because these bacteria live inside squids and fish in small organs and in symbiosis. So the squid gives nutrients to the bacteria, and the bacteria produce life for the squid. So they are located down here, so that the squid, when it goes hunting, does not produce any shade and can go hunting. So they thought, okay, this is very unique. It's not a common mechanism. Bacteria cannot communicate like that. Only these symbiotic vibrio can do it. So for many years, we still thought that bacteria were very boring. No communication, no coordination. Since the luciferase genes were very interesting as reported genes for molecular biologists, they were among the first genes that were cloned early in the 80s. So when they cloned the genes, they also cloned the genes that were involved in the regulation of the system. So this is the Lux operon, these are the Lux genes, and upstream we have the two proteins that are involved in the regulation. So LuxI is the protein that synthesizes the signal. And LuxR is the transcriptional regulator that senses the signal. So when you have low cell density, both genes are transcribed at a very low level. Right? So very low amount of signal is produced. And it dilutes away from the cell. So the signal cannot be sensed by the receptor, which is cytoplasmatic. But when the cell density increases, there's no change in the transcription of the genes, but an increase in the signal concentration. So the receptor can sense the signal and change the conformation, and it will bind a specific sequence that is upstream the operon that is called Lux box. And then the transcription occurs and light is produced. And at the same time, there is a loop that increases the production of the signal and the production of the receptor. That is why this phenomenon was called at that time self-induction, auto-induction. So it's a very curious phenomenon, but nobody thought it was important. Just very particular thing. And things took around 10 years to change. And it was absolutely by chance that in 3 laboratories simultaneously they discovered that this same mechanism was working in 3 very important bacteria. So 10 years later, in England, they discovered that the production of antibiotics was controlled by a similar system, production of a sensor by an homologous of Lux I, and the sensing by an homologous of Lux R. The same for conjugation in Enterobacterium and for the production of virulence factors in Pseudomonas aeruginosa. So when this was demonstrated, everybody thought, oh, maybe more bacteria communicate as we thought. So they started to look for signals in all the bacteria. At that time, they changed the name from auto-induction to quorum sensing. Quorum sensing means that the bacteria can sense whether enough number of bacteria are close so they can coordinate and launch a coordinated action. The signals are the same family of molecules. They are xylomosary lactones. Each bacteria produces different lactones depending on the substitution of the third carbon and the length of the lateral chain. So they can detect their signal or very similar signal, but not the signal produced by all the bacteria. And the number of bacteria that have that mechanism, gram-negative bacteria, is huge. So far more than 100 species are known to have this mechanism to control their gene expression. And some of them are very important. One of them is Pseudomonas aeruginosa. It's one of the top pathogens that we have now in hospital. And in this species and in many other species, virulence factors are being controlled by quorum sensing. So that means that bacteria coordinate to attack us as a coordinated army. Okay? So this is the signal that is being used by gram-negative bacteria to coordinate, but now we know that there are other signals, other languages. Gram-positive bacteria use these small peptides. I'm not going to go in detail about the gram-positive signaling. It's very interesting. And there is another signal which is called autoinductor 2, or AI2, that is being used both by gram-positive and gram-negative bacteria. That is what is called multilingual bacteria, and I'm going to show you just a little bit about AI2 because my talk will focus on acetylmonoamine lactose, gram-negative communication. And the story of autoinductor 2 discovery is very nice. All that work was done by Bonnie Basler back in the 19s with one vibrio. The vibrio you've been seeing is Vibrio fischeri. There's another vibrio, marine vibrio, that produced light called Vibrio harvegi. Vibrio harvegi was not behaving the way Vibrio fischeri was doing. So the system seemed to be much more complicated. So they knew that Vibrio harvegi had AHLs, but there were more signals involved. So she created a lot of mutants to try to find what are the other signals involved in the production of light and Vibrio harvegi. And while checking that, he had this mutant that cannot sense AHLs. It's called Vibrio 170. And when she tried, this arrow, double arrow here is Vibrio harvegi, the mutant. And she faced that to Salmonella and E. coli and see what happens. She wanted to know if those bacteria could produce some signal that could be sensed by Vibrio harvegi. So when she turned out the light, this is what she saw. E. coli and Salmonella were producing a signal that was stimulating light from the mutant. So there was a signal that could be sensed by many other bacteria and produced by many other bacteria, and it serves for bacteria to communicate among them, not only within the same species. I'm not going to go into detail about the quorum sensing channel of Vibrio harvegi because it's very complicated. It's very similar to a very important pathogen in bacteria, which is Vibrio cholera, human pathogen. Just very shortly, because you will need to have an idea of this to understand some of the results I will show later. In Vibrio harvegi, there are 3 different quorum sensing signals. So you have AHLs, AI2, and a third signal that is close to fatty acid. And in this case, the sensors are not intracellular sensors. They are sensory kinases that are on the membrane. Okay? So when no signal, low cell density, what you have here is a phosphorylation cascade, you know very well, that end up with the production of small inhibitory RNAs. These RNAs inhibit the expression of general regulators. Okay? But when you have the signals, then you have kinase, and this just looks O, which is a description regulator, is phosphorylated, is dephosphorylated, and then the inhibitory RNAs are not produced. The result is the expression of LOXR and the expression of quorum sensing target genes. That includes live genes in Vibrio harvegi and all the virulence factors for Vibrio cholera. And this system, LuxPQ and LuxO, is present in many bacteria, gram-positive and gram-negative. And in some of them, it controls very important factors. For example, in Escherichia coli. Escherichia coli does not produce AHLs. So it does not have a looks I, looks R system, but it has a looks S, looks OP system. So it produces and senses AI2 and controls a lot of genes. A total of 400 genes are upregulated or downregulated by this signal. This includes virulence factors, very important virulence factors. Interestingly, this is AI2. The E. coli sensor can be activated by this molecule that you can see here. It's very similar to AI2. That molecule is epinephrine, adrenaline, is our stress hormone. That means that E. coli can sense our stress. In the same sense, at the same time that he is sensing how many E. coli are around, so we can launch an attack and make this guy become sick. Right? So before talking about how we can take benefit of this bacterial communication, I would just make a very short comment on quorum sensing in cyanobacteria. Since there's a lot of researchers here, molecular biologists working on cyanobacteria, I just wanted to tell you a little bit about what we know about quorum sensing in cyanobacteria. When I first entered quorum sensing, I came from different cyanobacterial laboratories, so the first thing I did was to check if these cyanobacteria were producing AHLs. And we got very disappointing results because the AHLs, sometimes they were there and then they disappeared. What we know is that cyanobacteria have enzymes to degrade the quorum signal. So in some senses, they can sense it, and AHLs affect very strongly nitrogen fixation in cyanobacteria. We didn't go any deep on that in our laboratory, but there is a very well-known quorum sensing mechanism in cyanobacteria, and it's related to the differentiation of heterocysts in nitrogen-fixing filamentous cyanobacteria. So for those of you that are familiar with that, you know that many filamentous cyanobacteria can develop specific cells that are in charge of nitrogen fixation. So photosynthesis and nitrogen fixation cannot happen in the same cell simultaneously because nitrogenase is extremely sensitive to oxygen. Okay? The problem for the cell is that it is differentiation. So differentiation has to be very well coordinated. How to decide which cells will turn into heterocysts and which cells continue doing photosynthesis. And cyanobacteria can decide that very well, and in a very, very precise way. So they have a pattern. 10 to 12 cells are doing photosynthesis and one heterocyst. Another 10 or 12 cells and one heterocyst. So how do cyanobacterial cells know if they have to differentiate to heterocyst or not? Well, they are using a quorum sensing mechanism, which is not very well known, but I will explain. So this is the current model we have. There are two genes that are heavily involved in heterocyst differentiation. The first gene is the regulatory genes called NTCA, and you probably know that gene is a sensor for nitrogen deficiency. So in this cyanobacteria, the NITC will activate the expression of FR. FR is the key regulator for heterocyst development. But FR will not be active if we have a signal in the culture media called PAT-S. So, yes, the culture media. PAT-S is produced by the cells at the same time that FR, and diffuses along the filament. So close to the filament, the concentration is close to the heterocyst, concentration is very high. But far from the heterocyst, the concentration of PAT-S is very low. So when the concentration of PAT-S drops below the limit of detection, then that cell will differentiate as an heterocyst. The way that PAT-S interaction with FR is not very well known, but it is clear that it's through inhibition of their activity. So it's an inhibitory signal. It's a very small peptide, and it works exactly the way that many small peptides in gram-positive bacteria do. Okay, so that is just to make you to get you an idea that quorum sensing is also working in cyanobacteria. Here you have expression of FAT-S. FAT-S is being produced only in the telcis and diffuses along the filaments to inhibit the production. So let's come to the application. Why is this important? I'm not a molecular biologist. I'm a biotechnologist. So for me, the important thing of a process is what can we produce with it? How can we can take benefit? And for me and for everybody that was working or was aware of quorum sensing in 10 years ago, it was very clear. If bacteria only attack us if they are communicating, if we interfere with the communication, they will not attack us. So we have a chance to treat microbial infections by inhibiting quorum sensing. And then this is a demonstration with Pseudomonas aeruginosa. This is a mutant that does not communicate, does not produce the signals. And this is biofilm formation. So this is wild type. You see the Pseudomonas is producing a film that is covering the pink surface. That is the way it colonizes and creates infection in hospitals. But the mutant produces very weak biofilm. So it's much more easy for our immune system and for antibiotics to act on this than on this. So at that time, some 10 to 12 years ago, many pharmaceutical industry companies and many laboratories got interested in finding ways to interfere with quorum sensing. So that is my subject today. Quorum quenching as human pathogenics. And of course, when men think of a possible molecular mechanism, nature has already thought of it much earlier. So we already have examples in nature of inhibition of quorum sensing, which is called quorum quenching. This is an algae, red algae from Australia, and it was known a long time ago because it has very low bacterial colonization on the surface, right? And more surprisingly, almost all the bacteria that could be found on the algae were gram-positive. And in the seawater, we almost have no gram-positive bacteria. So some people thought, oh, maybe the algae is producing quorum sensing inhibitor. So what you see here is that they put the algae on a plate of a bacteria called Chromobacterium violaceum. So Chromobacterium violaceum produces this purple pigment when it's communicating. So you can see here very clearly that the algae is inhibiting the communication in the bacteria. How to do this? Cheating with the bacteria. So it produces this molecule. It's very similar to a furanol. So it's blocking the receptor of the bacteria, so the bacteria doesn't know that there are other bacteria around and will not form biofilm and will not colonize the algae. The inhibition of quorum sensing has been called quorum quenching, and there are two different mechanisms. The production of inhibitory substances that block the receptor, or the enzymatic degradation of the signal. The enzymatic degradation is much more extended than the production of inhibitors, right? And even we humans have enzymes that can degrade the signals and try to avoid infection like that. And regarding so there are two types of enzymes that are able to eliminate the signals. One are called lactonases. We have a type of lactonases in our blood and in our lungs. And that they open the lactone ring. And acylases that remove the fatty acid tails from the molecule and inactivate the communication. Okay. So very early, some 8 years ago, we had the first demonstration that quorum quenching could work to prevent infection in animals and plants. But we don't know yet, it hasn't been tested yet for human beings, but yes, we have enough demonstration for animals and plants. This is inhibition of infection by a Vibrio strain in troots, in fishes. So the troots were infected with the fish, and at the same time they were given furanones, the substances, the small substances that are produced by the algae. So you can see that almost all fishes died when no furanone was supplied. But if you supplied relatively small amount of furanone, you could inhibit the infection. Fish would not die. So the only problem with this is that fish were not dying with infection, but they were dying on the long term with the furanone, because furanone is extremely toxic. Another demonstration was made with transgenic plants. So these plants were transformed with a lactonase from bacillus. Is the best known quorum quenching enzyme. And you can see here the expression in different mutants of tobacco and potato plants. And here is the sensitivity of infection with Burkholderia, which is a plant pathogen. So on the top you have plants that have quorum quenching capacity and plants with no quorum quenching capacity, and this is the difference in the susceptibility to infection. So the presence of a lactonase was protecting the plant from infection. So that was the time that we entered the quorum sensing world, and at that time most people were looking for inhibitors of quorum sensing and bacterial strains that could inhibit quorum sensing in terrestrial habitats, in soil. And they could find almost only bacillus. So since we were marine biotechnologies, we thought, okay, quorum sensing was discovered in the sea. We have a huge diversity of bacteria in the sea, and we should go and see what kind of quorum quenching activities we can find there. And there was another reason, additional reason for looking in the sea. That's because most of the fishes that you eat and have been cultivated will die because of infection with gram-negative pathogens that use quorum sensing signals. And the problem with aquaculture is that you cannot use antibiotics. This is strictly prohibited in Europe, probably also in Japan. So you cannot, if you have a farm, a fish farm, and the fish get sick, there's nothing you can do, they will die if you don't have an alternative for antibiotic treatment. So what we do, we took the long way. Most people working on soil microbiology, they thought we will take the enrichment shock. So they wanted to isolate bacteria that could eliminate the quorum signal. So they used cultures that only had quorum sensing signals as a source of carbon. And they always ended up with bacillus. So we said, okay, instead of that, we will make a collection of marine bacteria, very different marine bacteria, from very dense microbial communities, the surface of an algae, sediment in tanks, different tanks and on beaches, and also from water, from coastal water and open water in the Atlantic Ocean, and see what happens. So we create a collection of more than 600 isolates, marine isolates. This is how they were distributed. And start the screening, functional screening. So one by one, those 600 strains were checked for the ability to interfere with the communication. And we did that with biosensors. Biosensors that were produced by the laboratory in Nottingham, Professor Miguel Cámara, which we collaborated with. And some of the first biosensors we tried were Lux biosensors. E. coli that has been transformed with a plasmid that has the Lux operon with the sensor, but obviously it does not produce the signal. So we expose the signal to the marine bacteria and then check the activity on the biosensor. And we got very, very high amount positive. 31% of our strains were positive, and 2 of them have had wide spectrum activity. That means it could interfere with the activity of almost all the signals that we were trying. That was very strange. So when we went to check, chemically analyze what was going on with the HL, that was false positive. That's because the Lux biosensors are very, very sensitive to other types of inhibition. So we were not seeing exactly quorum sensing inhibition, but just light production inhibition. So we had to change the sensor. And we came to the common bacterium biolastic sensor. So it takes much more time, and it was much more robust. So what we did, you have here plates that are covered with the sensor. The sensor does not produce the signal. You have to add externally the signal in these wells. And when the signal has been distracted or inhibited by the marine bacteria, you have no pigment production. Here is control or a bacteria that doesn't have quorum quenching activity. So pigment means no quorum quenching activity. No pigment means quorum quenching activity. And it's a very robust system because you can very easily detect and differentiate when you are having growth inhibition and when you're having quorum sensing inhibition. Okay? So here you have very common type of growth inhibition, which is swarming behavior in marine bacteria, and which usually gives false positive in different types of sensors. Of course, the activity had to be changed to chemical methods to be sure that we were having real interference with the shells. All the strains we got, they had enzymatic activity. None of them had inhibitory activity. And that was the summary of the result. And it was very surprising. Around 50 to 20% of our strains had some kind of quorum quenching activity. In soil, only 2% of the strains had quorum quenching activity. So that means that in seawater, the activity is much, much, much higher than in soil. But the activity is not the same in every marine sample. So there are samples which are almost very similar to soil. For example, water from coastal water. But open ocean water has very, very high activity, almost 25%. And bacteria that grows on the surface of this algae, almost 50% of the bacteria have the capacity to interfere with quorum sensing. So that really confirms that bacterial interaction in seawater seems to be much, much more active than in the soil, at least for those bacteria that we can cultivate. Because then the problem became, so what we are looking is at the activity in cultivated bacteria, but you know that in seawater, we can cultivate less than 1% of the bacteria that are present. So what happens in the whole picture? We see all the bacteria. So to do that, you go to look into the metagenomics, marine metagenomics, and look at the sequences. So we try to make a collection of all the known sequences of acylases and lactonases, and check for the presence in marine metagenomes. And we got some very curious results. Gauss collection, if you know the Gauss collection, were the first metagenomes that was created by Craig Venter. In Gauss collections, the presence of quorum quenching enzymes is low. But if you check the other collections, it's much, much higher, including soil metagenomes. That means probably that there is a strong differentiation in the type of bacteria that are present, the non-cultivated bacteria. And how abundant are these genes in comparison to other genes? So we check the frequency in comparison with other genes, very common genes, phosphate transporter, ammonium transporter, quorum quenching genes, and this is degradation genes. So you can see quorum sensing gene, quorum quenching genes are as abundant in metagenomes as almost ubiquitous genes like ammonium transporter and phosphate transporters. So that means that there is a lot of quorum quenching activity going on in the sea. But why? Because the concentration of bacteria in the sea is very low. So it's not possible that the concentration of AHLs reached very high levels. So there are two hypotheses to try to explain this. First hypothesis is that there is a lot of quorum sensing and quorum quenching activity in the sea, but it's not in the seawater, but in the very small particles that are called marine snow that are present in the sea. That is one. And the other one is that really this picture is not showing us real quorum quenching, and because we don't know if the quorum quenching enzymes are specific for quorum quenching, or maybe they're just more general degradation enzymes that are being used to degrade other type of molecules. So we're working on that now, but just very briefly show you the results from the biotechnological part. So from all our collection of bacteria, there are 90 isolates that have a very wide spectrum of activity. 2 of them have been patented. Some of them are completely new bacteria. For example, isolate 139 shares only 94% identity with all known bacteria. These have been patented, and for those of you very young people that are here, and probably, I don't know if that's the case in Japan, but in Europe, when you do apply to research, a university and government will very much push you to go into patent work. So you have a result that can be applied, you have to patent. Please consider patenting, because your research will change very, very much after patenting. So many things that you would like to do, you will never do. And many things you do, you cannot tell, or you can publish for a long time. So every day, since I patented this, every day when I get up, I regret doing it. But that's the way governments make us work. So one important thing about these strains is that they're taxonomically very diverse in comparison with what we know about soil. So for soil, which is in black, you see there are many concentrated in the beta and gamma proteobacteria. But the red are the marine. You can see we have very few in the proteobacteria, quite a few in the alpha proteobacteria and gram-positives, and they're not included here, but in the open ocean we have a lot of bacteroides, which are very unique bacteria. So it seems that the diversity in the sea is much, much higher than the diversity in soil. Just a couple of slides to show you how active are these bacteria. So this is a bacterial consortium with quorum quenching activity. Those are marine bacteria. And this is how fast they can eliminate the signal. So these are different consortiums, and the fastest consortium can remove completely the signal after 48 hours. But 20J, which is one of our patented isolates, can remove it similar concentration only after 30 minutes. Much faster. And which is very important for the biotechnological application. You don't need the live cell to do that. You just need a pure an cell extract can do that. So the enzyme is active, even if the cell is not alive. No specific cofactor requirement. So the enzyme is much more active in seawater than in PBS, and it takes only 60 minutes to completely eliminate the signal. So for normal scientists, the next step would be to clone the gene, and overexpress the enzyme and characterize. But since I have patented, then I have to spend 2 years doing crazy things, like the company will ask you, I want to know exactly how many kilos of bacteria I will need to treat 20 kilos of fish or something like that. And you have to make crazy experiments that at the end make sense, more or less, because they will tell you what is the chance that your product will be in the market. So probably we have now enough indication that we're going to have quorum quenching products in the market. Why? Because the companies think that if a microorganism is to be put in a feed, or a component of the microorganism has to be put in some product or material, you cannot go higher than 10% biomass. That means 10 grams per kilo. So how much biomass you need of this bacteria? Get activity 100 milligrams per kilo per liter. That means that companies may consider using it. If you go over 10 grams, then it's not commercially feasible. But of course, while you do this, you still have time to make some experiments. And here you can see the effect on the expression of virulence factors in the fish pathogen. The fish pathogen is Bacillus tapa. And this is the growth curve. The evolution of the signal in a culture of this pathogen. If you add the enzyme, the extract, to the culture, you can see you can completely reduce the production of the signal in the culture. So that causes very important changes in the behavior of the pathogen. So we are now working with a group of fish pathogens in Spain that are checking what are the changes in gene expression when you add this. And of course, we also could find the time for cloning the gene. It was hard because standard techniques did not work. So PCR strategy did not work. Standard primers for lactonases and acylases did not work. So we constructed a plasmid library, genomic library of our strain 20J, and used this selector strain, which is called quorum inhibitor selector 2. This is a Pseudomonas aeruginosa that has been transformed with a selective gene called levosucrane, SACB. I don't know if you're aware if you know this gene. This gene is toxic, so the expression of this gene kills the cells. So when you have HLs in the culture media, this gene was being expressed, and only if you have inhibitor, the gene will be silent and the cell will survive. Still, no result, no positive clone. So we had to go to functional screening. I really hate functional screening because I had a very, very bad experience with functional screening when I was young. But we had to go back to it. So we prepared a phosmid library of our clone. Phosmid library can contain fragments of 40 kb, kilobases. And so that would allow you to screen a much lower number of clones to have a chance to get your gene. So we screened around 7,000 clones, and we got one single positive clone. This one. This is expressing the quorum quenching gene. We sequenced the fragment. It's a long fragment, so we have to use different strategies, pyrosequencing and subcloning in an E. coli plasmid library, and we finally got the sequence. So it's a lactonase, and it has the typical same binding domain of beta-lactonases, but it has a very, very low homology with non-lactonases. So maximum is 38% with lactonase reactivity to the pathogens. That also has a very important ecological consequence, because the search that we made in metagenomic were used with these lactonases, terrestrial lactolysis. But then you get a marine lactonase, and it's completely different. So that may mean that we have more quorum quenching enzymes in the sea than we thought. So that's the result we have predicted. And now we are overexpressing to be able to characterize the enzymes. Just almost finishing. Most important thing is, okay, we have an enzyme. In vitro, we can inhibit infection or inhibit the expression of virulence. But can we cure infection in vivo in animals? So that is called proofs of concept. You have to prove that what you have is working, and we do it in aquaculture, in fishes. So we are at the moment testing if we can put this enzyme in feed and give it to the fish when they're sick to cure them. And also as a probiotic, because small fishes do not feed on inert material. They feed on small animals. And so you have to put live bacteria in the culture so that they can feed. So very recently this year, it has been demonstrated that you can use quorum quenching to cure fish. This is an infection of zebrafish with Aeromonas hydrophila, and they feed the fish with bacillus enzyme, this bacillus enzyme. And you can see here that you can protect the fish by reduce the mortality by 60% by feeding the enzyme. We have been doing, we are doing these experiments with fish, and we are also doing experiments with clams and oysters. The culture of clams and oysters, when they're very small, almost microscopic, is very difficult because they're extremely sensitive to the presence of bacteria. But you cannot use antibiotics to treat the bacteria in your culture tanks. So the idea was to put our quorum quenching bacteria in the culture tank and see if we could protect our little clams or little oysters from infection. So that's what we got. I will just show you one figure. This is an oyster, cellulose. It's survival after 20 days, which is a critical period for survival in oysters. This is survival in control. When you add antibiotic, it's well known that if you add the antibiotic to the larvae, it will improve the health of the larvae, so you will survive. Survival will increase. But if you add the probiotic, the quorum quenching strain, the result is even better than with the antibiotic. Okay? No result if you use very low concentration, but it gets concentration. So we have a good chance that we can use it to protect our animals. One very surprising result. In marine water, Vibrio are most dominant strain. And when you play the Vibrios, you see yellow. So this is a standard culture, marine culture in a culture facility. It's full of Vibrio. If you add antibiotic, no Vibrio. But if you add the quorum quenching strain, no Vibrio. Why? Because the quorum quenching strain is not killing the Vibrio. It doesn't have antibiotic activity against the Vibrio. It's just interfering with the quorum sensing channel Vibrio. So what's happening is that by interfering with the quorum sensing channel Vibrio, is decreasing the fitness of the Vibrio in the culture. So the Vibrio is expressing less enzymes, has less capacity to compete with other bacteria that are present in the water. So the number of Vibrio is decreased almost at the same level as by using chloramphenicol. Just to finish, very short thing to explain that sometimes you have very hard times in science, but sometimes science gives you very, very high satisfaction without looking for it. So parallelly to the aquaculture applications, we have been working on applications in dental health. Why? You know that dental plaque is one of the most complex microbial biofilms that we have. And many of the bacteria present there cannot be cultivated. Right? And the presence of that biofilm is the origin of different diseases, caries, and periodontal disease, gingivitis. For caries, you have mainly gram-positive bacteria taking care of that. But for gingivitis, it's mainly gram-negative and many bacteroidetes involved. So since we have been working on bacteroidetes, and we have some work that I'm not presenting on the production of AHLs by this type of bacteria, we were working on, first of all, we have difference in AHLs in dental plaque between sick people and healthy people? And can we isolate bacteria with quorum-quenching activity from patients that are always healthy, that never had any dental problem? So while working on that, results I'm not going to show, and to do that we collaborated with a researcher in Valencia called Alex Nia, and we sent some samples of our 20J quorum-quenching strain to check if the strain could inhibit the production of dental plaque. So he prepared the plates, and there were some empty holes in the plate, and he said, what about if we try Streptococcus and Staphylococcus? And I said, but those are gram-positive. I do not expect any activity on gram-positive bacteria, only on gram-negative bacteria. He still did, and he got this. So this is normal growth of Streptococcus mutans, which is the main actor in the formation of dental plaque. If you put extracts of 20J, this is furanol, inhibitor of quorum sensing activity, AHLs, for gram-negatives, and you see some kind of reduction. But if you use 20J cell extract, 90% reduction in the production of biofilm. So 20J is interfering with the quorum sensing signal, probably, we don't know yet. But our idea is that when 20J is affecting quorum sensing signaling in Streptococcus, which is a gram-positive, and that of course was not expected. We checked, it's not affecting growth. It's just affecting biofilm formation. So Streptococcus produces gram-positive peptide signals, but it also produced AI2. Remember AI2 from biblioharvey? This signal here that is produced by Lux S. So our hypothesis right now is that 20J is able to interfere with the quorum sensing mediated by AI2 in Streptococcus mutans. So to do that, we checked with some biosensors that were created by Bonnie Basler on biblioharvey, and this is the result. So wild type can sense the three signals. This appears in the first line here. BB170 cannot sense AHL, only AI2 and Chi1. And the third biosensor, third column, can only sense AI2. And what you check the production of light. So this is control. This is after addition of 20J. You can see that in all cases, the addition of 20J reduces or delays the production of light. That means that 20J is also affecting the communication channel that is mediated by AI2. We still don't know if it's through the same mechanism, the same enzyme that is affecting the AHLs, or if it's a different mechanism that have both activities in the same bacteria. So just to finish, because it's late and we're tired, what do we expect in the coming years from quorum sensing and quorum quenching? I'm quite confident that we are going to have these quorum quenching products in our pharmacies maybe in 10 years. we will be applying quorum quenching enzymes to a wide range of applications. First of all, I'm sure that we will still discover many new signals and functions of quorum sensing. In dental plaque, in intestinal microbiota, not so long ago, the complete metagenome of intestinal microbiota was released, and that will surely help us to understand the microbial interactions in such complex environments where most bacteria cannot be cultivated, and probably cyanobacteria. So cyanobacteria have enzymes to protect themselves from AHLs. So there should be some signaling of AHLs in cyanobacteria. Very important field, quorum sensing in microorganisms, in eukaryotic microorganisms. Some fungi use quorum sensing signals, not the same as bacteria, different ones. They're very relevant from a clinical point of view. I'm very sure that we will have the development of new pathogenics for treatment of human infections and novel antifouling materials based on quorum quenching. And of course, there's a very active field of research to understand what is the ecological significance of this signaling bacteria and interfering with communication. Just to finish, introduce my collaborators. So external collaboration with Professor Miguel Cámara from the University of Nottingham. That was the first laboratory in Europe working on quorum sensing. And Alex Mira is working on metagenomics of dental plaque. The laboratory of Professor Rodríguez Valera in Spain, they're exploring metagenomic. And Alicia Santoranto, she's a fish pathologist, and is helping us to elucidate the effect on expression of virulence factors and some pathogens. And our team, there's Manuel Romero, the work I'm presenting is mostly his PhD thesis, and Celia Meyer and Andrea Muras are PhD students. And these are the microalgae team, because we still have some microalgae in the lab, and they collaborated in the experiments with larvae of clam and oyster. And that's all. Thank you very much for your attention.